Picture this. You're walking into your Cambridge Biology 9700 exam. You flip the page, and boom, you see a massive 6-mark question sitting there on 4.2 active transport. Specifically, it's asking about endocytosis and exocytosis. Now, if you're like most students, you immediately start furiously writing down every single fact, every definition, and every biological pathway you've ever memorized from your textbook. But here's the thing. Right there, before you've even finished your very first paragraph, you've likely made the single fatal error that literally costs critical grade boundaries. So in this explainer, we're going to dive deep into 4.2 active transport, focusing on endocytosis and exocytosis, and we're going to unpack the high-mark exam strategy. We're going to expose exactly what that cold open mistake is, and more importantly, how we can apply the precise framework that senior examiners are actually looking for.
Okay, let's tackle this core problem right out of the gate. Why do students find multi-step biological pathways so incredibly hard? I mean, is it because the biology of active transport is just impossibly difficult to understand? Well, no, not really. It's because when candidates face an exam question on endocytosis or exocytosis, they panic. They instantly revert to these totally generic answers. Basically, they completely ignore the specific biological context of the question. You literally cannot just regurgitate a textbook definition of vesicular transport and expect to get top marks. These pathways are highly dynamic, and your answers, well, they need to be just as dynamic and tailored to the exact specific scenario the examiner has placed right in front of you.
And to really raise the stakes here, you have to hear this direct warning straight from the Cambridge Principal Examiner report. They stated, quote, "Answers on 4.2 active transport are often too vague. Examiners want specific details," end quote. The theoretical architecture of this entire topic rests on precise definitions, clear causal relationships, and integration with other areas of the syllabus. The assessment framework rigorously, and I mean rigorously, prioritizes analytical depth over superficial breadth. Look, simply stating that exocytosis moves things out of the cell, that's superficial breadth. Detailing the exact mechanism of exocytosis for, say, digestive enzymes from pancreatic cells in a given experimental case study, now that is the analytical depth they are actually demanding. So, what's really fascinating about these top 3 failure points is how clearly they map out the classic traps for the 9700 syllabus. Number 1, as we just saw, is those vague answers lacking specific details. Number 2 is forgetting to check units and terminology. Remember, practical work in this topic develops experimental skills, and that requires highly accurate observations and data recording. If you miss the units on a graph about active transport rates, you just lose the mark. It's that simple. And number 3, providing answers that aren't specific to the given context. Top-performing students exhibit this incredibly connected, conceptual understanding that they can transfer to totally unfamiliar contexts. If your revision strategy is just, memorizing flashcards without practicing application, it's absolutely going to fall short here. Okay, so you might be wondering, how do we actually secure these marks when the clock is ticking and the pressure is on? We use a real game changer called the method mark strategy. This strategy is all about earning exam marks for demonstrating the correct procedural process, even if, and this is key, even if your final conclusion or calculation ends up being totally flawed. When you get hit with a highly complex data analysis question about active transport rates or vesicle formation, the examiner really just wants to see your logical, structured reasoning. Your process is honestly often just as valuable as your final answer. So, how do we actually put this into practice? We use a standardized 5-step procedural framework for tackling these questions logically. Let's break it down. Step 1, identify the key elements. You need to deconstruct the question and extract the relevant data from the stimulus materials. Step 2, apply the relevant principles of active transport. Step 3, execute your analysis with super clear working and correct notation. Step 4, verify those results and spot any weird anomalies. And finally, Step 5, present your findings in the appropriate format using the absolute correct terminology. Think about it. If you make a tiny arithmetic error in Step 3, but you clearly applied the right principles back in Step 2 and used the right format in Step 5, the method mark strategy ensures you still bank those crucial points. Now hold that thought, because in just a moment, I'm going to reveal a hidden examiner clue that literally changes your answer completely. It's this massive realization that shifts you from just getting average marks to hitting that top-tier bracket. But before we get to that, we really need to quickly understand the technical analytical framework that makes this hidden clue so incredibly obvious in the first place.
Right. Let's briefly shift our focus to a critical technical area, statistical analysis in active transport.
This right here is an area where an enormous number of candidates drop points simply because they pick the wrong analytical framework. Practical questions aren't just about describing what happens, you have to prove it statistically.
You absolutely must map exactly which statistical test belongs to which data application so you're always applying the right tool to the evidence. For example, if an experiment is comparing the mean rate of exocytosis between two different sets of temperature data, you're going to use the t-test. If you are analyzing categorical data, like, say, the presence or absence of specific transport proteins, you use chi-squared. And if you're trying to find relationships between variables, like the concentration of ATP and the overall rate of active transport, you deploy correlation tests. Nailing the right test is step 1 of that method mark strategy we just talked about.
Let's do a quick, interactive pause and predict challenge. I want you to play the examiner for a second and spot the error. Imagine a student writes an answer explaining the process of a cell taking in large molecules via endocytosis. Their answer is absolutely packed with memorized definitions. They state all these generic facts about the cell membrane folding, but they don't include a single unit from the provided graph, and they completely fail to mention the specific cell type that was named in the prompt. So, what is fundamentally missing from this student's answer? Take a mental guess right now.
All right, let's see if you hit the nail on the head. This perfectly illustrates the gap between a mid-tier and a top-tier candidate. The flawed answer relies entirely on memorized definitions with generic facts and missing units. It's just a brain dump. But look at the top-band answer pipeline. You start with a concept, you back it up with evidence and data, and that leads directly into scientific reasoning tied explicitly to the context. A top candidate identifies the concept of endocytosis, extracts the precise data from the stimulus material, and then uses scientific reasoning to explain exactly why this specific biological pathway is happening in this specific scenario. It's a seamless, totally logical flow of information.
Now, we need to escalate to those higher-order assessment objectives. Consider this a mini comparative analysis masterclass. When an exam asks you to evaluate or compare approaches, let's say comparing active transport with facilitated diffusion, you have to be rigorous. You must explicitly identify similarities and differences. You have to use comparative connectives in your writing. You must support every single comparison with hard evidence from the provided data. But honestly, even that isn't enough. To snag those final top-tier marks, you need to draw justified conclusions about relative importance and explicitly identify limitations, assumptions made, or even alternative interpretations and how they affect the conclusions. That is what true analytical depth actually looks like.
Which brings us right back to that open loop, the ultimate examiner clue I promised you earlier. Here it is, loud and clear. Examiner alert. The classic mistake is providing generic answers, not specific to the question context. Always link every point to the given scenario. The hidden clue is, and always has been, the context itself. Principal examiner reports continually highlight that candidates who reference cross-topic connections and tie their conceptual understanding directly to the unfamiliar context provided in the exam achieve significantly higher marks. Context is not just fluffy background information, it is the absolute core requirement of your answer. We have covered some incredible ground in this explainer today. We've got the method mark strategy down, we know exactly which statistical tests to apply, we understand the concept to evidence pipeline, and we know for a fact that context is king. But wait, what happens if you misread the command word that dictates the entire mark scheme? What if the question asks you to evaluate, but you only describe? Well, unfortunately, this entire beautiful analytical framework we just built collapses instantly. If you don't know how to decode specific Cambridge command words, you are going to lose marks, even if your biological knowledge is 100% perfect. Decoding those command words is the absolutely vital, unresolved next step for your exam success, and it's exactly what we are going to be covering in our very next explainer. You seriously cannot afford to miss it. So to make sure you don't fall into that trap, subscribe to our YouTube channel, Ace the Exams at acetheexams_IGCSE to excel in your Cambridge examinations. Master the strategy, understand the context, and I will see you in the next masterclass. Until then, ask yourself, the next time you look at a multi-step biological pathway on an exam paper, are you just seeing memorized facts, or are you finally seeing the underlying data waiting to be decoded?